Yeah, so today I wanna tell you something about invasive interfaces, but in this case, interfacing the brain, the nervous system, to connect external devices. So it's another acronym BCI, but it's actually brain-computer interface. So you have to remember one acronym for multiple devices. So it's also called like field of neuroprosthetics. So neural prosthetics or prosthetic connected with the nervous system. I just founded the Neurobionics lab here, where we are trying to create this neuroprosthetic device, starting from the mechatronics part, so the electronics and the wearable device, that part that is actually actuated and is used to interact with environments. But also we study the neural interfacing, so how we can connect this device with the nervous system, with the peripheral nerves, with the brain. And in particular, the topic of today will be about intracortical brain-computer interfaces. So those type of devices that are connected directly to the brain, to the cortical regions, in this case, for the restoration of sensory motor functions in people with paralysis that can be spinal cord injury, ALS, or amputation in more extreme cases. So we are part of a consortium called Cortical Bionic Research Group. Is a consortium between American universities and now also Chalmers, so the University of Chicago, Northwestern University, University of Pittsburgh, where actually there is a knowledge of how to do this type of implants, so and in particular inpatients. So there are 5 participants chronically implanted, the one up to 10 years now with this type of device that is working with us in the lab. So since we wanna, so the goal of the Cortical Bionic Research Group is to restore sensory motor functions, in particular related to the upper limb and arm and hand function in particular. You need to remember and consider that when you want to replace a limb or the function of a body part controlled by some neural mechanism, it's very important to know how the biological counterpart. So you want to recreate a robotic limb behaving in the same way as your arm. And so you need to know and understand very well how the natural system, the biological system, is controlling this arm and this part of the body, because it's very useful since you are interfacing a robotic device with the nervous system. It's not just a robotic device that you can control using your algorithms and your other intelligence, but you need to use the same principles that are used in the human body in order to interface in the correct way. So we start, since we want to restore the hand function in particular, we need to think that the human hand is pretty complex in terms of neural representation. So in your brain, the control of your hand is not, it's pretty complex. It's multiple degrees of freedom. It's actually one of the 2 flagships of humans. So it's the hand, and the structure of our cortex is actually the thing that distinguishes us from many other animals. And you do like these simple tasks that you do in daily life that are also peculiar of humans in terms of intelligence. So you express your intelligence through your manual behavior. And these are pretty complex, but in daily life used without any type of problems in reality, right? But if you want to replace this with a robotic device, with a prosthetic device, it's pretty complex to do. So you extract, when you interact with objects, a part of the control that you had the opportunity to see during this workshop, you have a very important part that is the somatosensation, and the part that is extracting the information from the object that you are touching in real time, few milliseconds, and you're able to extract a lot of information of the object that you are touching without even thinking about it. But you know the shape of the object that you are touching, the temperature, the texture, and all these properties. And interestingly, you use also your hand differently accordingly to the feature that you want to extract from the object. No, you squeeze the object to understand the stiffness, or you move the hand on the surface to understand the texture. And so you are exploiting your biological sensors. So you have biological sensors in your hand that you use to extract this information. So what happens is that you have your larger organ in your body, so your skin, that is mechanically deformed, and this is creating a pattern of skin deformation, mechanical. And then you have these biological sensors that are able to convert this mechanical deformation in pattern of electrical activation, that is the language of the nervous system. So if you stick an electrode in the nerve, when you're touching something, you will be able to see a specific pattern of activation inside your nerve, specific from the object that you are touching. And if you go up on the neuroaxis and you implant an electrode in your brain and you measure again the electrical activity there, you will see another pattern of activation of this part of the cortex that is again encoding all the information of your interaction with the external environment. So there is a specific coding of the object that you are touching. And from that coding, your brain extracts the perception. So what you are actually feeling, so the texture, the shape, and all the properties. So a little bit of background of the somatosensory system. So as I said, we have these mechanoreceptors. So these biological sensors in your hand, you touch something, the signal is converted to electrical signal into your nerves. The nerves goes to the spinal cord, and then depends by the part of the body, for the upper limb to the cuneate nucleus, and then to the thalamus, and then goes up to the cortical regions, so the primary somatosensory cortex, where actually the basic features of, similar to V1 for vision, are extracted to understand edges, motion, and properties of the thing that you are touching. And then the signal goes in higher order, secondary somatosensory cortex, and so on, where actually the perception is integrated with your memory, with other senses, etc. So in particular, you have these small biological sensors in your skin that are extracting the information of the object that you are touching. So in particular, you have Merkel discs, highly densely populating your skin, that when you touch something are extracting the coarse feature of the texture that you're touching. So this is giving back to your brain a kind of texture image of the thing that you are touching, spatially distributed. So you can see actually the coarse part of the texture that you're touching. And then when you slide your finger on a surface, you have other type of receptors, the Meissner and Pacinier corpuscles, that are encoding the vibration of your skin when you're moving on a texture. So this is giving the temporal code of the texture that you are touching. And so together with the spatial part extracted from the first type of mechanoreceptors, you have a spatiotemporal coding of the texture that you're touching, and you're actually feeling this in your brain. So there is a specific temporal and spatial coding in your nervous system. For example, here you see a case where if you touch an object and you actually pick and lift this small object, you have the load force, the grip force, and so you can relate these forces that you're applying on the object that are translated into your skin with a specific pattern of activation of these mechanoreceptors and these fibers that are encoding the contact, sliding, lifting, force. Everything then is perceived by your nervous system. So then you have different type of receptors, as we said, that are activated differently because are encoding different information when you touch an object. So this just to say that the nervous system is encoding with complex pattern, spatiotemporal pattern of neural activity, the mechanical interaction that you just had with environment. And in few milliseconds, really, you're able to adjust your motor control. So this signal is integrated then with your motor control in order to adjust the grip if it's not enough because the object is sliding and is falling from your hand. And so there is a very symbiosis and connection between the sensory and motor system. So this information is then projecting, as we said, a different level of the neuroaxis, different part of your brain, and depending by what type of interface you're using and what type of disease that can affect some of these pathways, you can interface the neuroaxis at different levels. We will see some examples. In particular, for the brain-computer interface, we go into the brain. And in particular, we go in a region called primary somatosensory cortex. It is a region close to the central sulcus, where actually the information are arriving from lower level processing, arriving into your brain. It's pretty superficial, so it's pretty easy to access if you open the skull. Is as some properties that we will see are crucial for the development of a neuroprosthetic device. And in particular here, we can, if we stick an electrode here and we measure the activity, we can actually see the activation and the properties of the object that we are touching. One example is that, so if you apply an edge on your fingertip and you measure the activity in the primary somatosensory cortex, and you can see here that if you change the orientation of the bar on the fingertip, the population, the neurons that you are recording are responding differently and are encoding, in this case, the orientation of the edge that you are touching, because are responding more for this orientation, less for this other orientation, and together with other population of neurons are making the network aware about which orientation is with which orientation this bar is deforming the skin. And so we can extract, for example, a tuning curve. So the response of the neurons that we are recording in the brain accordingly to the orientation of the bar on the skin. And this is valid also for direction of motion, etc. So just to tell that these neurons are actually responding accordingly to that. This was just a quick overview about the somatosensory system. So we know that is important. The code, of course, our nervous system is encoding information, and spatiotemporal information are pretty important. In case of, so there is a very famous experiment performed by Professor Johansson here in Sweden, proving very simply how much these sensory information are important for motor control. So here you can see a subject, an healthy individual, performing a task like lighting a match. Pretty simple, few seconds, no problem. So here you use vision, perception, touch when you do this. And then after the anesthetization, so you anesthetize the tip of the fingertips of middle finger, index, and thumb, and you ask the healthy subject to perform the task again. So in this case, the subject is struggling to pick this small object. You can imagine that if you have there is a cold day and your fingers are very cold and you try to do something very fine, this becomes difficult. And here vision is clearly not able to replace touch. It's not suitable to extract this tactile information from the environment with the same speed and integrate with the motor control. And even after training, so if you do this multiple times, you are still worse than in case of natural touch. And this is the case of people with some type of disability or disease or injury. Here is an example where if I ask you what is wrong in these people, you would probably tell me something related to the motor system. So it's like, this is like a neurological condition affecting the muscles. But in this case, these people are completely healthy in terms of motor functions. They have a rare neuropathy where they have lack of sensory, they have lack of big diameter fibers in their nerves, so related to the proprioceptive and tactile, so these biological sensors that we mentioned before. And as the lady without with anesthetization, they are not able to do or very slow to accomplish simple tasks. So when we do artificial touch and we want to recreate this sense and embed this in a brain-computer interface, we need to have, if you think about the robot, the sensing part as our skin, we need to replace that. We need to create the pattern of activation if we interface with the nerve through electrical stimulation, in this case, neural interfaces, so interface the nerve and activate, or direct interface the brain and then create the artificial perception in this case. If you have a question, of course, stop me. So there are different types of neural interfaces and electrodes, so tiny structures that you can use to record and stimulate the nervous system. Today I will focus more on the brain, so the central nervous system, but we have many types of interfaces developed also here at Chalmers by Maria Asplund, for example, tiny stretchable electrode implant that can be implanted in the peripheral nerves. For the cortical implants that you can use for a BCI, depending by the invasiveness, of course, of the device. So closer you are to the source that you want to record or stimulate, you are more invasive, of course, but also more selective. So you are able to exactly target the nervous tissue and the areas that you want. And in our case, what we decided to use are intracortical microelectrode arrays. This is the famous UTA arrays. Is a silicon-based electrode with around 100 tips that are normally insulated, and then you have just the tip exposed. And so when you deliver current from this micrometer tip, you can activate a group of neurons around the tip. At the same time, if you record from this electrode, you can actually measure, here you can see the electrode, and you can see the action potentials of the single unit that we can record from that specific part of the brain. Then are connected with a cable to a pedestal, this connector that you can see here on the anchor to the skull, and then from the skull is connected outside. So we want to create a bidirectional brain-computer interface or bidirectional bionic device. So what we do is that we need to interface this with a part of the brain that is encoding the motor intention of moving the arm. So this is the first side of the bidirectional device. So you need to implant, in this case, the primary motor cortex, where the movements are generated, and then you need to decode this pattern of activity and actuate a robotic device to do the movement. At the same time, you will record the sensors that you place on the robotic device, touching an object. You convert these sensors in pattern of electrical stimulation following the code of the natural touch that you saw before. And you can evoke sensation if you stimulate directly the primary somatosensory cortex. So which type of patients are suitable for this type of device? So we know that people with tetraplegia or paralysis due to the spinal cord injury, we cannot interface them into the nerves or muscles because the lesion is blocking the signal going up and down. So the only way to target is the brain, and the cortical areas are the best in terms of accessibility and also properties that we will see. People with tetraplegia in particular, they have as a highest priority to restore, to have back hand functions and arm functions, then sexual function, bowel function, and bladder function, others. So we have two ways to do that, or we try to reactivate their own arm or we use an extracorporeal arm. So we replace the arm, not physically, but just we attach a side of a wheelchair, another arm, and we allow them to interact with the environment through that arm. So how we do this? So we implant microelectrode arrays into this is the central sulcus, this is the primary somatosensory cortex, primary motor cortex. These are the arrays that we implant. So we implant 2 motor arrays in the arm and hand representation. So we are able to capture the neurons responsible for the control of the arm and the hand. And then we implant other 2 arrays in the representation of the hand. So to have sensation, so if we stimulate with these arrays, we can evoke vivid sensation perceived on the paralyzed hand. Then, so this is, then we have a robotic arm aside, controlled directly with these signals. So how we can do that? So we know that for the motor control, we have, so when you play the sound of the neurons in your primary motor cortex, when the monkey is moving the arm, the neurons in your primary motor cortex are firing. So you can actually record this with our microelectrode arrays. And what we know is that from a lot of non-human primates work, we know that if we move the arm, in different direction, population of neurons are activated differently. This is a reaching out task where if you move your arm in this direction and you record from this population of neurons, you will see that these neurons are responding more for this direction of movement in space and less for these. Similarly to the sensory neurons that I showed you for the tactile. So actually also here we have specific pattern of neurons activated when the movement is in some direction. So actually this is a typical condition where, similar to muscles, you have electrical activity, pattern of electrical activity, differently according to the movement. So you can use machine learning methods to decode the motor intention. There are some examples where, so here, for example, we use the velocity of the different joint, the hand, the shoulder, because in this case, we want to have a complex 10D, because you have also shoulder, elbow, hand. It's pretty complex. But there are now, I don't want to talk about the motor part, but we have other methods, more sophisticated, that are trying to, instead of using firing rates, trying to use how the population of neurons are responding, and measure more the pattern in the network in terms of trajectories. These are called manifold analysis, where actually your neural space is doing specific trajectories in your brain, and these trajectories are different accordingly to the movement that you do. So you can actually decode all this information to control the arm. And in this way, the patient is able to control a robotic arm with the brain and interact again finally with the environment. Or we can also enable the patient to use a virtual arm in virtual reality. So here you can see our participant with the headset controlling a virtual arm in virtual reality with the brain control, and do some task of reaching and grasping. And then, so what we add is the sensory part. So we have sensors on the robotic hand that we read in real time while the subject is touching object. We convert these sensor readouts in patterns of microelectrical stimulation, and we stimulate the correct part of the brain. How we can do that? We know that our primary somatosensory cortex, so where we place the electrodes, is organized, is called somatotopically. So that means that every part of your brain every part of your body is represented in your brain. So if you move inside the somatosensory cortex, okay, here you can see the gyros, and if you move here, okay, you will have different parts of your body represented in your brain. So if you stimulate, this is the work done by Penfield in 1950, where they map the homunculus, is called, so that your body in your brain. And more sensitive is the area of your body, larger space is represented in your brain. So more neurons are dedicated to that specific area. As you can see, the hand has a pretty big part of your brain in the somatosensory cortex. So that makes us able to place electrode directly in that region in order to target the hand, the fingertips. So what we ask the subject to do is we put, before the surgery, we put the subject into the MRI scan, and we ask the subject to move, imagine to move his thumb, imagine moving the index, and then we highlight in the brain the area, sensory motor areas related to the hand. Then once we know that, we take our arrays, and even if the gyrus, as you can see, the structures of the brain are a little bit different among our participants, at the end, we implant the electrodes always in the area where there is activation for the hand, so the primary somatosensory cortex. So after this implant, we start to stimulate. So what we do is a phase called projected field mapping. So that means that if you stimulate from one of our, these are like the two arrays there in the brain, and then we stimulate from electrode 1, and the subjects report some sensation, if is feeling or not sensation, and where exactly. So they can mark on the hand the part of where the sensation is felt. So we know that the neurons that we are activating through the stimulation of that channels are evoking a sensation on that patch of skin. So that is the projected field of that channel. So if we change electrode, we can change finger. If we go proximal, like in this direction is lateral, so lateral anterior, we move from the thumb to the pinky. So after that, we know that if from the different channels, we can evoke sensation on the fingertip. And again, we know how big is each finger in the brain. We have different maps for different participants because the implant can be not exactly in the same position for each participant. But the point is to evoke sensation on the hand. So when you stimulate, the subject can tell you where the sensation is felt, but not only, can also describe more. Because compared to all the experiments in monkeys, these experiments in humans allows you to have a description of sensation. So you cannot ask the monkey, what do you feel? And wait for reply. You can do psychophysical experiments, but in humans, they can actually describe what they are feeling. So then when we characterize the map of sensation with a correct sensation, we put this in the robotic hand. So that means that we have sensors in the hand. Every time that we touch the robotic hand index, the subject is feeling the sensation on the index. And so we can connect the brain and the robotic device.
Index, brain, pinky.
So here you can see my colleague at Pittsburgh, Rob Gaunt, touching the robotic hand in different fingers. The subject is blindfolded, and the subject is able every time to report which robotic finger is touched, because he's feeling directly the sensation on the hand.
Index.
Then this is the first step, so the contact. So we encode the contact of some object with the robotic hand. Another thing that you can do is to change all the stimulation parameters that you have. And after a lot of studies, we understood that if you increase the amplitude of the stimulation, you recruit more neurons with your stimulation, and so the sensation is perceived as more intense. So you are cramp up the juice, and the sensation is becoming more intense. We characterize that in three patients here, and you can see that if you increase the amplitude, these are different channels, different colors of different patients, more or less, you have an increase of sensation while you're increasing the amplitude. This is important because when you put this in the robotic hand, you can connect the pressure of your sensor. Higher is the sensor value, higher can be the amplitude that you deliver, and so the subject can perceive a stronger sensation. Then when you incorporate this and you ask the subject to do a task, you will see the same result as what was visible for healthy participant, the restoring sensation after anesthetization. So you have, in this case, the robotic hand brain control with the feedback, and at the right, you have without feedback. So in many different conditions with different objects. So what changed in this control in particular was the fact that the subject was immediately feeling when the object was correctly grasped, how much force, and they moved much faster one object from one point to another point. So the control became more dexterous. But if you compare the best brain control arm, robotic arm with feedback with our hand, still very bad. I mean, you can say, oh, that's cool, but then, I mean, if you consider the amount of things that you can do compared to that, is pretty limited. So we started to try to go beyond the contact and force feedback, trying to study a little more about the sensation, because the sensation, we believe, is clearly the game changer for our ability to manipulate objects. So when you stimulate in your brain with imagine that you deliver current, if you deliver a stimulus like that, the subject will feel a sensation increasing, stationary, and then decreasing, no? But if you indent your finger, so with something that is pressing and releasing, the brain response, so you can see the raster plot here of the neurons in your primary somatosensory cortex, are much higher during the transient phase than during the sustained phase. So that means that the neural activity is completely different from this pattern that we deliver, because you have higher activity during the transient and lower activity during the static phase. So instead of sending a shape of stimulation more like this, you should send something more what we call biomimetic. So something that is mimicking more what your brain is doing. So we compare these two, linear versus biomimetic, and we observe improvements. I don't want to go into the detail, but I can give you, if you want, more sensitive feedback. So the sensation became more salient and more precise. The ability to discriminate force increase, and the number of level of discriminable force that you can distinguish increase remarkably compared to non-biomimetic approaches. Finally, I want to show you very quick something more, because your sense of touch is not only force and contact, but is actually able to extract all tactile features like shapes, motion, curvature. And then we said, okay, we need to find a way to encode this sensation in a natural way through our neurostimulation. How we can do that? First of all, first step is to study how the brain is doing that. So there are a lot of monkey work of looking at how your brain is representing the neural features of orientation, the neural feature of motion in your primary somatosensory cortex. So what we did is to start a very simple task. So we took our subjects and we have our map of projective fields that we saw before. And if you zoom in some of the projective fields, you can see that the subject is reporting specific regions on the finger when you stimulate with specific channels. So our idea was, would it be possible to stimulate with different channels at the same time and evoke a line projected field on the skin that can evoke a sensation of something oriented in a specific way. And this is what happened when you combine projected field in a aligned way on the finger, what the subject is reporting.
Okay, so this is more of a vertical, but it's diagonal. It's going from the pinky side to the thumb side on an angle. That's my finger.
So the subject is able to tell exactly how this sensation is evolved, where exactly this sensation is. So we create different orientations and we provide the subject randomly this sensation, and the subjects were able to tell the direction, the orientation of this stimulation on multiple digits of thumb, index, and middle finger. We replicate in a second participant as well. Another very trivial thing, if you think now, before was not, was like, why you don't activate a line projected field, but one after the other with specific temporal ordering to see if this will become a congruent unified sensation moving on the fingertip. And this is what happened.
Running on these two fingers, my middle finger and my ring finger, from the tips of the fingers to the middle of the knuckles, running in this direction. So running from the fingertip down towards the palm, doing it at the tip to the middle of my finger.
But it's movements from right here on the index finger, so that's the pinky side, going towards the tip and the thumb side. So it's going from right here on the side up to the tip of the finger, like halfway through.
So the subject was able to tell the different direction of motion reliably, and we replicate with another subject. I will skip this 1 second. And after that, so we characterize, we encode also other type of tactile features, and then we place everything into the robotic hand and see and design a closed loop task with this more complex sensation. In particular, what we did is to make the subject control a robotic arm steering a wheel. Okay, so with brain control. So we decode the brain intention of steering one left direction or stay still through the brain activity. Then we create a perturbation, virtual perturbation. So that means that we give a sensation of something moving in one direction or other direction. Imagine that you drive on the street and you feel a bump and your steering wheel is turning, and then you have to adjust to stay still. So you feel the motion and you adjust with your motor control. And this is like placing closed loop. So here there is the participant, and there is the robotic arm that the perturbation is created, and the subject has to move in the other direction. So we did this to prove that we also cognitive load, to prove that the subject was able to use this more complex sensation in a natural way and in a closed loop task. So for concluding, there is if you're interested in the state of art of BCI, there is a paper published recently in Nature Review Bioengineering, explaining more about the history of BCI with how many patients now are implanted around the world, which are the companies doing that. Of course, the neurotech market is growing pretty fast. There are more and more companies doing that, mostly in the US, but is becoming more and more a thing. You probably heard about Neuralink from Elon Musk. They have, for example, 2 participants implanted with a wireless system with thousands of channels that allows the patient with spinal cord injury to control a computer. And they're using this in daily life. There is an estimation of in a paper showing that these implants will become also more a clinical practice for neurosurgeons around 2035, like 2 implants per month per neurosurgeon. This is in the US. And just to mention, I want to show that the patient that I show you in the videos are our participants in Chicago. They are not completely paralyzed. They have partial spinal cord injury, so you saw that they can move. They decided to participate in this because they really want to help people even more impaired for the next generation of BCI. So they totally know that they will not benefit with this technology, but they want to help the others. So they are really the pioneers in that. And I want to just give you like a short video where Scott, our participant, is reporting why he's participating in this.
Scott and Bruce have been working more than 2 years. You have been a subject of this work for years now,
Yes, sir.
and I wonder why.
I wanted to have someone else have the opportunity to become independent again.
The most meaningful work of your life.
Yes, sir. 100%.
So they are very, very great people. So thank you very much. These are all many people are involved, and I'm open for questions.